Locomotion and movement. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movement. Streaming of protoplasm in the unicellular organism like amoeba is a simple form of movement. Movement of cilia, flagella, and tentacles are shown by many organisms. Human beings can move limbs, jaw, eyelids, tongue, etc. Some of the movement is result in a change of place or location. Some voluntary movement are called locomotion. Walking, running, climbing, flying, swimming are all some form of locomotory movement. Locomotory structures need not to be different from those affecting other type of movement. For example, in paramecium, cilia help in the movement of the food through cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for changes in body postures and locomotion as well. The above observations suggest that movement and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movement but all locomotions are not locomotions. Method of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic conditions, or to escape from enemies, predators. Type of movement. Cell of human body exhibit three main type of movement, namely amoeboid, ciliary, and muscular. Some specialized cells in our body like macrophages and leukocytes in blood exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of the protoplasm as in amoeba. Cytoskeleton elements like microfilament are also involved in amoeboid movement. Ciliary movement occur in most of our internal tubular organs which are lined by the ciliated epithelium. The coordinated movement of cilia in the trachea help us in removing the dust particle and some of the foreign substances inhaled along with atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. Movement of our limbs, jaws, tongue, etc. Require muscular movement. The contractile property of muscles are effectively used for locomotion and other movement by human being and majority of the multicellular organism. Locomotion requires a perfect coordinated activity of muscular, skeletal, and neural system. In this chapter, you will learn about the types of muscles, their structure, mechanism of their contraction, and important aspect of the skeleton system. Muscle. You have studied in chapter 8 that the cilia and flagella are the outgrowth of the cell membrane. Flagellar movement helps in the swimming of the spermatozoa, maintenance of water current in the canal system of the sponges, and in locomotion of protozoans like euglena. Muscle is a specialized tissue of a mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscles. They have a special property like excitability, contractility, extensibility, and elasticity. Muscles have been classified under uh, different criteria, namely location, appearance, and nature of regulation of their activities. Based on the location, three types of muscles are identified. First is skeleton, second visceral, and third cardiac. Skeletal muscle are closely associated with the skeletal component of the body. They have a striped appearance under the microscope and hence are called striated muscles. As their activities are under the voluntary control of the neural system, they are known as voluntary muscles too. They are primarily involved in locomotory action and changes of the body postures. Visceral muscles are located in the inner wall of the hollow visceral organ of the body like the alimentary canal, reproductive tract, etc. They do not exhibit any striations and are smooth in appearance. Hence, they are called smooth muscles, non-striated muscles. Their activities are not under the voluntary control of the neural system and are therefore known as involuntary muscles. They assist, for example, in the transportation of the food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of the heart. Many cardiac muscle cells assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature as the neural system does not control their activities directly. Let us examine a skeletal muscle in detail to understand the structure and mechanism of contraction. Each organized skeleton system muscle in our body is made up of a number of muscle bundles or fascicles held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contains a number of muscle fiber, figure 20.1 each. Muscle fiber is lined by a plasma membrane called sacrolemma. Enclosing the sacroplasm, muscle fiber is a syncytium as the sacroplasm contains many nuclei. The endoplasmic reticulum, example, i.e., sarcoplasmic reticulum of the muscle's fiber is the storehouse of calcium ion. A characteristic feature of the muscle fiber is the presence of a large number of parallelly arranged filament in the sacroplasm called myofilaments or myofibrils. Each myofibrils has alternate dark and light band on it. A detailed study of myofibril has established that the strange appearance is due to the distribution pattern of two important proteins, actin and myosin. The light band contains actin and is called I band or isotropic band, whereas the dark band called A or anisotropic band contains myosin. Both the proteins are arranged as rod-like structures parallel to each other and also to be longitudinal axis of the myofibrils. Actin filaments are thinner as compared to the myosin filament, hence are commonly called thin and thick filament respectively. In the center of each eye band is an elastic fiber called Z line which bisect it. The thin filament are firmly attached to the Z line. The thick filament in the A band are also held together in the middle of this band by a thin fibrous membrane called amyline. The A and I bands are arranged alternately throughout the length of the myofibril. The portion of the myofibril between two successive Z lines is considered as the functional unit of contraction and is called a sarcomere, figure 20.2. In a resting state, the edges of thin filament on the either side of the thick filaments partially overlap the free ends of the thick filaments, leaving the central part of the thick filament. The central part of thick filament not overlapping by thin filaments is called the H zone. Structure of a contractile protein. Each actin thin filament is made of F filamentous actins helically wound to each other. Each F actin is a polymer of a monomeric G globular actins. Two filament of another protein, uh, tropomyosin, also run close to the F actin throughout its length. A complex protein troponin is distributed at regular interval on the tropomyosin in the resting state, a subunit of a troponin masks the active binding site for myosin on the actin filaments. Each myosin thick filament is also a polymerized protein. Many monomeric protein called monomerasins, figure 20.3b, constitute one thick filament. Each meromycin has two important parts, a globular head with a short arm and a tail, the former being called the heavy meromycin HMM and the later the light meromycin LMM. The HMM component, i.e., the head and short arm, project outwards at a regular distance and angle from each other from the surface of a polymerized myosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding site for ATP and active sites for actin. Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of a muscle fiber takes place by the sliding of the thin filament over the thick filament. Muscle contraction is initiated by a signal sent by the central nervous system CNS via a motor neuron. A motor neuron along with the muscle fiber connected to it constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter acetylcholine which generates an action potential in the sarcolemma. This spreads through the muscle fiber and causes the release of calcium ion into the sarcoplasm. Increase in calcium level lead to the binding of a calcium with a subunit of a troponin on actin filaments and thereby remove the masking of active site for myosin. Utilizing the energy from ATP hydrolysis, the myosin head now binding to the exposed active site on actin to form a cross bridge figure 20.4. This pulls the attached actin filament toward the center of a band. The Z line attached to these actins are also pulled inwards, thereby causing a shortening of the sacroiliac, i.e., contraction. It is clear from the above step that during shortening of the muscle, i.e., contraction, the I band get reduced, whereas the A band retain the length figure 20.5. The myosin releasing the ADP and P1 goes back to its relaxed state. A new ATP bind and the cross bridge is broken figure 20.4. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation. And breakage is repeated casually further sliding. The process continues till the calcium ion are pumped back to the sarcoplasmic cisternae resulting in the masking of actin filaments. This causes the return of Z lines back to their original position, i.e. relaxation. The reaction time of the fibers can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to the anaerobic breakdown of glycogen in them, causing fatigue. Muscle contains a red colored oxygen storing pigment called myoglobin. Myoglobin content is high in some of the muscle which gives a reddish appearance. Such muscle are called the red fibers. These muscles also contain plenty of the mitochondria which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscle. On the other hand, some of the muscles possesses very less quantity of the myoglobin and therefore appear pale or visceral. These are the white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on the anaerobic process for energy. Skeleton system. Skeleton system consists of a framework of bones and a few cartilages. This system has a significant role in movement shown by the body. Imagine chewing food without jaw bones and walking around without the limb bones. Bone and cartilage are specialized connective tissue. The former has a very hard matrix due to calcium salt in it, and the latter has slightly pliable matrix due to the chondroitin salt. In human being, this system is made up of a 206 bone and a few cartilages. It is grouped into two principal divisions, the axial and the appendicular skeleton. Axial skeleton comprises 80 bones distributed along the main axis of the body. The skull, vertebral column, sternum, and ribs constitute axial skeleton. The skull is composed of two sets of bones, cranial and facial, that totals to 22 bones. Cranial bones are 18 in number. They form the hard protective outer covering cranium. For the brain, the facial region is made up of 14 skeleton elements which form the front part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity and it is also included in the skull. Each middle ear contains three tiny bones, malleus, incus, stapes, collectively called ear ossicle. The skull region articulates with the superior region of the vertebral column with the help of two occipital condyles, dicondylic skull. Our vertebral column, figure 20.7, is formed by 26 serially arranged units called vertebrae and is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebra has a central hollow portion, neural canal through which the spinal cord passes. First vertebra is the atlas and it articulates with the occipital condyles. The vertebral column is differentiated into cervical 7, thoracic 12, lumbar 5, sacral 1 fused, and coccygeal 1 fused regions. Starting from the skull, the number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, support the head, and serve as the point of the attachment for the ribs and the musculature of the back. Sternum is a flat bone on the vertebral midline of thorax. There are 12 pair of ribs. Each rib is a thin flat bone connected dorsally to the vertebral column and ventrally to the sternum. It has two articulation surfaces on its dorsal end and is hence called bicephalic. First, seven pair of ribs are called true ribs. Dorsally, they are attached to the thoracic vertebrae and ventrally connected to the sternum with the help of hyaline cartilage. The 8th, ninth, and 10th pairs of ribs do not articulate directly with the sternum but join the 7th rib with the help of hyaline cartilage. These are called vertebrochondral false ribs. Last two pairs, 11th and 12th of ribs, are not connected ventrally and are therefore called floating ribs. Thoracic vertebrae, ribs, and sternum together form the rib cage. Figure 20.8. The bones of the limbs along with their girdle constitute the appendicular skeleton. Each limb is made up of a 30 bone. The bones of hand forelimbs are humerus, radius, and ulna. Carpals, wrist bone are 8 in number. Metacarpals, palm bones 5 in number, and phalanges, digits 14 in number. Figure 20.9. Femur, thigh bone, the longest bone, tibia and fibula, tarsals, ankle bone 7 in number, metatarsals 5 in number, and phalanges, digit 14 in number, are the bones of the legs. Hind limb, figure 20.10. A cup shaped bone called patella covers the knee ventrally kneecap. Pectoral and pelvic girdle bone help in the articulation of the upper and the lower limbs respectively with the axial skeleton. Each girdle is formed of two halves. Each half of the pectoral girdle consists of a clavicle and a scapula. Figure 20.9. Scapula is a large triangular flat bone situated in the dorsal part of the thorax between the second and the seventh rib. The dorsal flat triangular body of a scapula has a slightly elevated ridge called the spine which protects as a flat expanded process called the acromion. The clavicle articulate with this. Below the acromion is a depression called the clinoid cavity, which articulate with the head of the humerus to the form of the shoulder joint. Each clavicle is a long, slender bone with the two curvatures. This bone is commonly called the collarbone. Pelvic girdle consists of a two coxal bone, figure 20.10. Each coxal bone is formed by the fusion of the three bones, ilium, ischium, and pubis. At the point of the fusion of the above bones is a cavity called acetibulum, to which the thigh bone articulates. The two halves of the pelvic girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. Joints are essential for all type of movement involving the bony part of the body. Locomotory movement are no exception to these. Joints are a point of contact between bones or between bones and cartilages. Force generated by the muscles is used to carry out a movement through joints, where the joint act as a fulcrum. The mobility at these joints vary depending on the different factors. Joints have been classified into three major structural forms, namely fibrous, cartilaginous, and synovial. Fibrous joints do not allow any movement. This type of a joint is shown by the flat skull bone, which fuse end to end with the help of a dense fibrous connective tissue in the form of the sutures to form the cranium. In cartilaginous joint, the bones involved are joined together with the help of cartilages. The joint between the adjacent vertebrae in the vertebral column is of this pattern, and it permits limited movements. Synovial joints are characterized by the presence of a fluid-filled synovial cavity between the articulating surfaces of the two bones. Such an arrangement allows considerable movement. These joints help in locomotion and many other movements. Ball and socket joint between humerus and pectoral girdle, hinge joint, knee joint, pivot joint between the atlas and axis, gliding joint between the carpal, and saddle joint between carpal and metacarpal of the thumb are some examples. Disorders of a muscular and skeletal system. Myasthenia gravis, autoimmune disorder affecting neuromuscular junction leading to the fatigue, weakening, and paralysis of skeletal muscle. Muscular dystrophy, progressive degeneration of skeletal muscle mostly due to genetic disorder. Tetany, rapid spasms while contraction in muscles due to low calcium in body fluid. Arthritis, inflammation of joints. Osteoporosis is related disorder characterized by decreases bone mass and increases chances of fractures, decreased level of estrogen in a common cause. Gout, inflammation of joint due to accumulation of uric acid crystal. Summary: movement is an essential feature of all living being. Protoplasmic streaming, ciliary movements, the movement of fins, limbs, wings, etc. are some form exhibited by animal. A voluntary movement which exhibit causes the animal to change its place is called locomotion. Animal move generally in search of food, shelter, mate, breeding ground, better climate or to protect themselves. The cell of the human body exhibit amoeboid, ciliary, and muscular movement. Locomotion and many other movements require coordinated muscular activities. Three types of muscular are present in our body. The skeletal muscles are attached to skeleton element. They appear striated and are voluntary in nature. Visceral muscles present in the inner wall of the visceral organ are non-striated and involuntary. Cardiac muscles are the muscles of the heart. They are striated, branched, and involuntary. Muscles possesses excitability, contractibility, extensibility, and elasticity. Muscle fiber is the anatomical unit of muscle. Each muscle fiber has many parallelly arranged myofibrils. Each myofibril contains many serial arranged units called sarcomere, which are the functional unit. Each sarcomere has a central A band made of a thick myosin filament and have two half I band made of thin actin filament on either side of its marked by jet line. Actin and myosin are polymerized protein with a contractility. The active site for myosin and resting actin filament are masked by a protein troponin. Myosin head containing ATPase and has ATP binding site and active site for actin. A motor neuron carries signal to the muscle fiber which generates an action potential in it. This causes the release of calcium from sarcoplasmic reticulum. Calcium activates actin, which bind to the myosin head to the form a cross bridge. These cross bridges pull the actin filament, causing them to slide over the myosin filament and thereby causing contraction. Calcium are then returned to the sarcoplasmic reticulum, which inactivate the actin. Cross bridge are broken and the muscles relax. Repeated stimulation of muscle lead to fatigue. Muscles are classified as red and white fiber based primarily on the amount of red colored myoglobin pigment in them. Bones and cartilages constitute our skeleton system. The skeleton system is divided into axial and appendicular. Skull, vertebral column, ribs, and sternum constitute the axial skeleton. Limbs, bone, and girdle form the appendicular skeleton. Three types of joint are formed between bones or between bones and cartilage. Fibrous, cartilaginous, and synovial. Synovial joint allow considerable movement and therefore play a significant role in locomotion. Thank you.